everybody. Today we're going to be answering the question, how do we build life? What are our little parts? And as you can see down below, the answer is biomolecules. So what we're going to do today is we're going to talk about those biomolecules, the different classes of them. Now, it's going to take some time, but you're in control of your pause button. So if you need to take a break, go ahead, take a break, hit pause, and then come back again. Let's get started. The molecules of life are going to be made of 4 different classes of biomolecules. First type, carbohydrates. Second type, protein. Third, lipids. And fourth, nucleic acids. Now, all 4 of these biomolecules have something in common, and that's the fact that they're all organic. So what that means, they all have a certain element in them, the element of life, and everyone knows it. That element is going to be carbon. So within cells, these small organic molecules can be joined together to form larger molecules, and often these carbohydrates, proteins, lipids, and nucleic acids, although the smallest little piece of them is very tiny, when we find them in the cell, they're usually huge in comparison to the little pieces that made them up. So we have a name for that, and that's going to be macromolecules. Now, I'll give you an example of a macromolecule. Here we have trypsin, and as you can see, there are a lot of different parts in this little thing. I want you to think of every little bulge you see in that picture as a different amino acid in a big protein called trypsin. Another thing I'm going to do today is, if you look down in the corner, if you see some of these little hearts that I put in the corner, I'm going to try to relate some of this information to things that will apply to the bigger picture of biology, or maybe human health. So, for example, trypsin. In the body, trypsin is a digestive enzyme that hydrolyzes protein. Its original molecule comes from the pancreas, and as food leaves the stomach and goes into the small intestine, one of the things that goes in there is the precursor molecule to trypsin, one of the molecules that helps hydrolyze protein or break down protein. To understand these biomolecules better, we need to be able to understand the difference between macromolecules, polymers, and monomers. So let's go ahead and take a look. First, a polymer is a long molecule consisting of many similar building blocks. So the easiest way I can think about it here is to put a train up on the screen. Now, the train is a big thing, and it's made up of very similar building blocks. And in this case, each train car is going to be one of those building blocks. So each train car, then, the small building block molecules, they're going to be called monomers. So in this case, there's 4 monomers to this polymer. And here's a monomer going away, there's another one, but the 4 of them together made up a polymer. Now, a polymer and a macromolecule are often very closely related, because once something has gained the status of being a polymer, once there's enough little tiny pieces in place, then you have a molecule that's big. Macro means large, so a large molecule. So polymers are often macromolecules. So I'll just go ahead and I'll throw an equal sign right there. The monomers then are the little pieces that make them up. Now, 3 of the 4 classes of those biomolecules or macromolecules are going to be classified as polymers. Carbohydrates, and what that means is you can have a lot of little ones that form something much, much larger. Proteins, and nucleic acids. So, by the process of elimination, which one is not qualifying as a polymer? And of course, the answer to that one would be lipid. There is a huge diversity of polymers that exist. Each cell will have thousands of different types of macromolecules within. So you can build a whole bunch of these different molecules from a very, very small set of monomers, those little tiny building blocks that build the biomolecule. So why is that? Well, first let's start off very basic. Let's get back to the element carbon. Carbon, since all of these particular biomolecules are organic, it means it's going to be present in every type. And as we know, carbon can bond to 4 different things. So if that's the case, you could have many different attachment possibilities. Here's carbon with its 4 hands that it can join up with other stuff. And if we get a little bit more scientific, here we can see carbon in its 4 valence electrons. So we'll go ahead and we'll imagine that we have some other atom coming in here to this place here, and I'll use a different color now. Something different here and something different here. So maybe the red and the green additional electron that are shared can swap places in a different molecule here or here. And if that's the case, you can approach a huge amount of diversity that's possible with all those enormous possibilities of bonding. So reason one then is versatility of the carbon atom. The fact that it has 4 attachment points allows for a lot of different possibilities. Second, functional groups. Functional groups are extremely important when we talk about biology because it leads to how certain molecules are going to react with other molecules. And those functional groups will also lead to functional diversity and molecular diversity. So here is a table showing our functional groups. Now, should you memorize all of these functional groups? Well, I'll say this. It does help. However, it's not necessary in an intro level bio to know exactly all of the chemistry of each one of these. Again, it does help, but if you're a little shaky in the chemistry, you can still survive without having a total grasp of the chemistry of how these work. But let's go over each one. The first type of group is called a hydroxyl group. Now, notice you see a little dash here, and what that means, that's a covalent bond, and you see an oxygen and a hydrogen that are connected to that single bond. Now, what goes here? Well, typically it's going to be a carbon atom. Now, we don't know if the molecule has 1 carbon atom or 15 carbon atoms, and the bonding spot might be on the 8th carbon. We're not sure, and that's why they leave it blank. Just like in this carbonyl group here, we could put a question mark here and a question mark there as well. Because typically what we're going to see is this is going to be part of a carbon chain. There'll be a carbon at the end of this and a carbon at the end of this as well. Now, if we take a look at where we find these functional groups, you can see that hydroxyl group are often found in carbohydrates. Carbonyl groups are often found in lipids. Something called a carboxyl group is a carbon double bonded to an oxygen and single bonded to another hydroxyl group right here. Something we're going to find a lot of when we study proteins. Amino groups, we see that there's a nitrogen atom with 2 hydrogens covalently bonded from it, and chances are, again, you're going to have a carbon atom connected to that open-ended covalent bond. And again, when we study proteins, we're going to meet a lot of amino groups as we look and see how proteins are built. And last but not least, from this table, we also see phosphate. You see a phosphorus atom, and you can see 1, 2, 3, 4 oxygen atoms around it, and you can also see a negative charge on a couple of those oxygens. And again, carbon, whether it's the end of a molecule or the middle of a molecule, either way could be possible in this location. Where do we find phosphate groups? We typically find them in DNA and ATP, and those are part of the nucleic acid family. At the start of the talk today, we talked about how these biomolecules had to be built, but they also can be broken down. So once you've made a very large thing, often biologically they need to be broken down again. So let's take a look at some of these ways that we can build and then break molecules. First of all, the type of reaction that it takes to build something up in biology is called a synthesis reaction. It's very common. More specifically, we're going to call it a dehydration synthesis. Now, I want you to think of the word dehydration. What do you typically think of when you think dehydration? What are you in dire need of? And the answer, of course, is water. So when water is lost from 2 monomers, what we're going to get is a link together. We're going to build that molecule bigger by losing a molecule of water. Now, we also call this, is also known as condensation. Both dehydration and condensation, hopefully the reference point to you will be that water causes both to happen. Okay, second, polymer breakdown. Polymers will be disassembled to monomers by hydrolysis. Okay, I broke the word up because hydro means water, lysis means breaking. So hydrolysis is water breaking. A reaction that's essentially the reverse of the dehydration synthesis reaction using the addition of water. So in the first reaction, when we build something up, water comes out. But in the second type of reaction, hydrolysis, what we see is water must go in to break down your bigger polymer. So it will facilitate the breakdown of polymers. Now, there's also going to be a cast of helpers that are going to allow certain things to happen in terms of the body faster, and those are called enzymes. Enzymes are protein macromolecules, they're helpers, that are going to speed up the dehydration and hydrolysis reactions. So if we take a look at this big purple blob down here, what we see is that we see a red molecule entering this space right here. When it fits in real nicely, something happens, and now the red molecule has broken into a blue and a green, and now they're released. And if you look at the place that allowed that to happen, it's right back to the way it was before it occurred. So we can have certain types of molecules be helped out in terms of their breakdown by these big purple blobs, which are protein and their enzymes. And what they're going to do is they're going to speed up this red molecule, whatever that is, breaking down. Now, the red might break down on its own, but with a big purple helper like this enzyme here, it's going to go even faster. Let's take a look at a picture as to how a molecule gets built. So right here we have a short polymer made of monomers 1, 2, and 3. It's a 3-piece polymer. The third piece happens to have a hydrogen atom hanging off the end of it. Now, we have an unlinked monomer, one all by itself over here, and it has a hydroxyl group, an OH, hanging off the side of it. Now, if this unlinked monomer heads this way and it's going to bang into the short polymer, what we're going to see is there's going to be a connection between this H and this OH. These three atoms have an affinity for each other, and what they're going to do is they're going to take off from this molecule. They're going to find each other, like each other, and then they're going to leave in the form of a water molecule. Okay, so here's our little water droplet. It gets pulled out, and what's left over then is going to be this stem here and this stem here. And if this continues to go towards one another, the stems will then connect at this location right there. So what we have here with the loss of a water molecule is a dehydration synthesis reaction. We're removing water, dehydration, to build something bigger. Because we had short polymer 1, 2, 3. We have now short polymer 1, 2, 3, 4 because we now have a connection point that got connected through the loss of the water molecule, making this polymer longer. So dehydration synthesis reactions lengthen polymers. We can also look at a picture to show the breakdown of a polymer. Here, we have the same polymer as last time. We have polymer 1, 2, 3, 4 that got built. But let's say there's a lot of water around, and that water is going to be on a crash course with that molecule 1, 2, 3, 4. It slams into it, and what happens is for a brief moment, there is a connection between whatever atom was sitting on the edge of this 3 and whatever atom was sitting on the edge of this one, number 4. And what happened is the original atoms went back to the same place they were before. So by the nature of the collision, maybe it hit just right at the right angle at the right speed. The H reconnects onto, in this case, monomer number 3. The hydroxyl group reconnects onto the unlinked monomer, which from up here was number 4, and it goes back to the way it was before. So this is an example of a hydrolysis reaction. Instead of taking a water molecule out, we're putting a water molecule in to break up a larger polymer. So hydrolysis of polymers break down polymers. So we've mentioned a lot of things building up and a lot of things breaking down. So imagine every time you eat, you're bringing in rather large food molecules, which are shown by these symbols right here. These large food molecules don't stay that way as they pass through our body. They go through what's called catabolic pathways. The word catabolic means to break something down. And when things get broken down, not only are we going to receive useful forms of energy like ATP and heat, but we're also going to get all of those very large food molecules will be breaking down into these small little parts here, the many building blocks for biosynthesis. We said synthesis was building things up. Because we're an organism in action that has a working metabolism 24/7, we need products built in our body every second of every day. So then these little tiny parts here are going to go through anabolic pathways, which are certain types of pathways that build up substances. So for this to happen here, we said that it needs to be broken down with the help of water. So hydrolysis is going to make this happen here, which are our catabolic pathways. Hydrolysis, there we go. And then to build those things back up, we're going to need dehydration or condensation reactions to happen. We pull the water back out and that builds things up different. And if you look at these things that get built up here, notice they are slightly different from what we ingested the first time. So they can reconfigure themselves and arrange themselves in different ways and combinations that we didn't see before, something that our body now needs. The first type of biomolecule that we're going to look at today, we're going to focus in on what's called the carbohydrate family. So these include sugars and the polymers of those sugars. Now, the very simplest molecules of this group are going to be called monosaccharides. So they are single sugars. And if we look over here on the right, we can see our most famous example of one of these, which is glucose. And right here, we have kind of an organic shorthand way of drawing a glucose molecule. Now, if you remember the functional groups, we said that hydroxyl groups would be found on carbohydrates. And look at all of them. There's an OH there, an OH there, an OH there, there, and there. There's a lot of hydroxyl groups on this thing. But we also said it was organic, which means that we have a lot of carbon in it. But I only see one carbon. Well, remember that organic shorthand, to make it a shorthand, means that we're not going to draw every single thing that's there. But what I need you to remember is that at every point where there's a bend in the structure, you're going to find a carbon there. So although you don't see them, there's a carbon here, 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 and there. So that makes glucose a 1, 2, 3, 4, 5, 6 carbon sugar, okay? And its formula is C6H12O6, okay? If you counted everything up, that's what you're going to come up with. All right, so our monosaccharides then are single, simple sugars. Macromolecules, on the other hand, are going to be polysaccharides. That means 2 or more of these monosaccharides are going to bond together to form a larger structure. So in the case of sucrose, which you can see to the right here, you have a 6 carbon sugar right here, but it happens to be bonded to a 5 carbon sugar right here, this upside down pentagon. This is called fructose. So here we have glucose and we have fructose coming together to form a molecule called sucrose, which is a disaccharide. So there's 2 single monosaccharides bonded together right in the center right here to make a disaccharide. Now, if we look at the bottom example, this is called amylose, and this is a polysaccharide / starch. And here you see a 6 carbon sugar, a 6 carbon sugar, a 6 carbon sugar. So this one is 4, but these can get hundreds to thousands of units long, going from monomers to very, very large polymers. So then simple sugars, which are also known as monosaccharides, they're going to have molecular formulas that are usually some multiple of the formula you see here. Look at the ratio of elements to one another. What's the ratio? So if you go back to your chemistry mind for a second and we look at this formula, how many carbons are here? Well, the answer is 1 because we don't see a subscript. So it's 1 to 2 hydrogens to 1. So monosaccharides have a 1 to 2 to 1 ratio. Glucose, C6H12O6, is the most common monosaccharide. And we know if we divided each of these subscript by 6, you'd get 1 to 2 to 1. So there you go. Fructose would be C5H10O5. So there you go, a 1 to 2 to 1 ratio again. Monosaccharides are also going to be able to be classified, and we do so by a couple of different methods. One is the location of the carbonyl group. Again, back to our functional groups, the carbonyl group has a structure that looks like this. You have a C double bonded to an O, which is bonded to an H, like that. So the location of it can either be on the end of a molecule or it can be right in the center. We can also classify monosaccharides based on the number of carbons that we find in their carbon skeleton as well. So with all this information out here, as of this point for carbohydrates, what do you think monosaccharides could be used for in animals and plants? And if you're thinking the fact that a sugar usually gives you energy, that's a big part of it. So storing these sugars is an important thing that living things do, because we have to have a constant source of energy at any given time, and we use carbohydrates as fuel. This table here is broken down a couple different ways. On the top here we can see trioses, pentoses, hexoses. What the heck does all that mean? Well, we all know what the prefix tri means. A tricycle has how many wheels on it? 3. So what we're saying is that this column here, these are 3 carbohydrates. So 1, 2, 3, 1, 2, 3. That's how we classify this particular group of carbohydrates. A pentose. We know what the prefix pent means. It means the number 5. So 1, 2, 3, 4, 5, and 1, 2, 3, 4, 5 for the ribulose down here. And then hexose, hexagon from geometry class, again, we know the number 6. So these should all be 6 carbon sugars. And if you count them all up, that's indeed what you will find. Now, on the left side here, we see a couple of weird looking words, aldoses and ketoses. And what they're trying to explain here is that this group here of glyceraldehyde, ribose, glucose, and galactose, they're all related because in each one of these, the carbonyl group is found at the end of the molecule. This is the carbonyl group right here, and you'll notice that it's in the same location in each one of these molecules. Where if something qualifies as a ketose, that carbonyl group then is going to be found in the center of the molecule and not at one of the ends. Now, this slide looks kind of scary, but don't let it scare you. We have colors to help us out, figure out what's going on here. So first, on the left here, what we have is we have a 6-carbon sugar. We see a lot of hydroxyl groups, so likely this is going to be one of our carbohydrates, and it actually is. It's glucose. And you'll see a couple of double-headed arrows right here, which means that they're in equilibrium. But what it takes for this molecule here to change into that is an aqueous solution. So when this straight chain structure here finds water in some way, what we're going to see is that that straight chain structure is going to fold itself up. And right here you can see hanging off the edge here we have a hydroxyl group on the 5th carbon. And what's going to happen is this H right here is going to go ahead and it's going to detach. And it's going to connect onto that O right there. And meanwhile, this H here is going to stay basically where it's at right now, but we're going to form a bond between this carbon and this oxygen, folding it up into a nice ring-like structure. So this is what glucose actually looks like when it's in biological fluid. It's in a ring-like structure. We mentioned how monomers join together to build polymers earlier, and that's through the loss of a water molecule. So the simplest type of polymer that you can make from 2 sugar monomers is going to be what's called a disaccharide. So through that dehydration reaction, we're going to join 2 monosaccharides. And if you check out this picture here, we're going to go ahead and add a glucose to a glucose. Now, on this carbon right here, there's a hydroxyl group. On this carbon right here, we have a hydrogen. And they are going to bang into each other, and they're going to briefly connect, form a water molecule, leave the area, joining these 2 monomers together. When a glucose and a glucose come together, we form the disaccharide maltose. Here you can see the O that is now in between the 2 of those molecules holding them together. Down below, we can see a different connection. We have glucose and we have fructose. Putting these 2 simple monosaccharides together through the loss of a water molecule, like we saw before, we're going to make the disaccharide sucrose, better known as table sugar. Now, that covalent bond through the loss of that water molecule actually has a name, and it's called a glycosidic linkage. When you see the prefix glyco, I want you to think sugar from now until the end. So if you see glyco, know it has something to do with sugar. Now, if we take a look at each one of those bonds, the first one on top is called a 1,4 glycosidic linkage. Now, what does that mean? Well, it just means that carbon number 1 on this glucose and carbon number 4 on this glucose are actually going to be linked in the center, and therefore it gets the name the 1,4 glycosidic linkage. Now, the glucose-fructose connection, however, is called a 1,2 glycosidic linkage. So in this case here, the fructose is numbered a little bit differently. This would be your carbon number 1 up there. Here's 2, here's 3, here's 4. So in this case here, this glucose, its carbon number 1 is bonding to this fructose's carbon number 2, and that's why it's a 1,2 glycosidic linkage. Now, through those dehydration synthesis reactions, we can form some very large, some very long chain sugars. And we mentioned before, we characterize these best as polysaccharides. They have storage and structural roles. So, what's going to happen here is that the function of this polysaccharide is going to be determined by whatever sugar monomers make it up and the position of the glycosidic linkages. They will matter with certain organisms in mind. The helical shape of this one here in glycogen is going to allow for enzymes to easily find locations for hydrolysis. Now, if you forgot what an enzyme was from a few slides ago, it was one of those big blobs. Usually they're drawn in purple, but what it does is it has a little active site and it allows for certain molecules to nestle into that little active site right there, and it allows them to split easier than if it was without that particular enzyme molecule. On this slide here, we're going to look at the difference in glucose storage from plants to animals. So on the left, hopefully you saw right off the bat that you have a chloroplast up here. So this would be which type, plant or animal? I'm going to trust you that you know that one. And here we see some starch molecules. Now, they just look like big white blobs when we look at this little image of a chloroplast. But if we go down below here to the artist rendering, we can see that we have a couple different types. We have one that's called amylose, and we have another one called amylopectin. So one of these here seems to be, it kind of goes in a helical form, kind of like an old phone cord. This one here has some branching coming off of it, but again, still the helical shape. Now, in this case here, this is typical for plants. So starch is going to be the energy storage molecule of plants. And we're going to find that storage molecule within the chloroplast of a plant. Now, on the right, we see something a little bit different. We see mitochondria, first of all, and we see glycogen granules. Now, if you look at the basic monomer here, we see the same type thing as we saw over here. We still see the kind of old phone cord typed helical shape. We see a lot of branching coming off of this one. And this is the particular storage carbohydrate of animals. And again, its name is glycogen. So where would you find glycogen? Well, let's think of what needs energy quickly. Your muscles do, okay? So we find a lot of glycogen stored in muscles. But we also have an organ, your abdomen, that stores a lot of it too when we need to increase the amount of glucose in our body and then restore it again. That organ is going to be your liver. So next we're going to look at the structural polysaccharides of starch versus cellulose. Now, these two polysaccharides are both found in plants. Now, they're going to be slightly different. What I want you to do for a second right now is take a look at this alpha glucose versus beta glucose. They both are going to originate from this 6-carbon chain sugar right here, this monosaccharide called glucose. But do you see any key differences between them? If you said the OH, you're right. That's a hydroxyl group. And in one of them, you'll notice the hydroxyl group is below the plane right here. But in the other one, you're going to notice the hydroxyl group is above the plane right here. How will that affect the structure of the polysaccharide? Well, we'll see it in just a second, but take a look down below at how they link up together. Notice that in starch, there's a 1,4 linkage of alpha glucose molecules, monomers. So in this case here, we see the oxygen in the middle. We know that to get that to happen, we had to lose an H2O in each of these connections right here through dehydration, also called condensation. But in the other picture over here, maybe you notice that the hydroxyl group here is below the plane, above the plane, below the plane, above the plane. Now, how would that happen? Well, the only way that would happen is if the glucose molecules got flipped the other way. So in this case here, we see the positioning of the hydroxyl group above and below, above and below. We still see the oxygen connection between the two monomers, but this one goes up, this one goes down, this one goes up, next one goes down. So the glycosidic linkages are going to differ slightly from one to another because overall, what we see in the one is that all the glucose line up in the case of starch, but in the other one we have kind of the up and down repeating pattern when we talk about cellulose. Now, what's the importance? Well, cellulose, first of all, is going to be a major component of the cell walls of plants. The starch, as we mentioned in the last slide, is going to be the energy storage molecule found in the chloroplast of a plant cell. In this slide here, we're going to take a look at the two different forms of glucose, and we're going to see how that affects the overall shape of a polysaccharide. So let's go back to the last slide just for a second so we can see the difference between beta glucose and alpha glucose. So in this case here, alpha glucose had the we had the hydroxyl group down below the plane. But in the beta glucose, we saw the hydroxyl group above the plane. So if we go back to this slide here, we'll notice that with the beta, we said that the glucose molecules will flip, one up, one down, one up, one down. And what that's going to do is going to produce some real straight chain looking polysaccharides there. So what we can say then is in straight structures, H atoms on one strand can bond with OH groups on the other strand. Where does that happen? Right here in this red circle. So not only do we have this straight line chain right here, but we also see connections between chains, which is going to make this a very strong substance, which we find again in cellulose, what we said was prevalent in cell walls. Secondly, the parallel cellular molecules held together this way are going to be grouped into what's called microfibrils, which is that strong building material. So this is one tough substance right here. Now, on the other hand here, we had the alpha glucose, the one where we had basically the connection that always went downward from the previous slide, and that's going to be prevalent in what we said here was amylose, the starch. And starch is going to be something that doesn't form these big organized sheets like that. Instead, it forms this helical shape, and this helical shape is susceptible to enzymes that do that hydrolysis thing we were talking about before, where if something needs sugar, you can break this bond here very, very easily, especially with the help of an enzyme, and there's no problem getting to the interior of the molecule. There's a lot of space there. So what we could say then is enzymes that digest starch by hydrolyzing alpha linkages can't hydrolyze the beta linkages in cellulose. So it's a big no for here. We can't go ahead and bust up those glucose molecules very easily, but we can do it here. And that's why we derive sugar from starch and not from the cellulose that we eat. So if we can't eat cellulose, who can? Well, the answer to that are microorganisms. So we don't do very well with cellulose. Cellulose is the type of sugar that we find in like grass and tree bark and all the things that really don't taste that good to us. But little microorganisms, it's absolutely delicious to them. So what they're going to do is they're going to hang out inside of organisms that give them some protection in what we call a symbiotic relationship. Let's take a termite, for example. A termite eats wood. We know that. So wood comes in, and then we have this wood fiber, but cellulose is indigestible, right? Well, if you look inside of its abdomen here, you notice there are protozoans. These are little tiny critters that live inside, and they can use that cellulose fiber, and they can turn it into glucose, which then is going to fuel this termite. So what does the protozoan gain? It gains protection by the termite. What does the termite gain? Well, it gains the ability to get energy from cellulose, which it normally couldn't do unless it had protozoans inside of it. Same thing goes for with a cow. A cow is supposed to eat grass all day long. Not all cows do, unfortunately, but they're supposed to. And when they do, it usually eats a lot of grass real fast. And when you eat a lot of bulk food really fast, the digestive system has a hard time with it. So what's going to happen is this grass is going to enter a chamber called the rumen, which is full of bacteria, which is going to mix and churn that grass into smaller and smaller pieces as it goes to work on that cellulose. The cow can then bring that and re-swallow that mass back up. So then the digestive system further on has a better chance and it has more time to extract the nutrients from it than if it just takes big masses of grass through the entire digestive system. Having that chamber there in a cow's rumen allows it to break down further. It can be put on the pathway then through the digestive system as smaller and partially digested, which makes the digestion of it easier. Alright, let's try to make sense of the big picture here. Down below you can see we have the beta glucose monomer, which is linked together hundreds and thousands of times to form cellulose. Now, That cellulose we said earlier are going to form little strands called microfibrils. Those microfibrils then are going to be part of the plant's cell wall, which we're going to find right here at the border between any two plant cells. So here's a plant cell there, here's a plant cell, there's one. So at all these borders, we're going to find these cellulose microfibrils making up the cell wall. And they are going to form what's the basic structure, in this case, of a maple leaf. Now, if you go outside and you grab a maple leaf and you start chewing on it, you're not going to get very much from that except a lot of insoluble material. We call that insoluble material fiber. So what passes through us is going to kind of do a little scrape job as it goes down through our digestive tract, especially through our intestines, and it's going to help clean out and keep our intestines healthy. That's why we're supposed to eat vegetables every single day. The last topic related to carbohydrates I wanted to talk about today is going to be a particular one called chitin. And as you can see above, this is a glucose molecule right here, but I want you to notice this blue box hanging off it. This is an amino acid. And what we're going to see is that we can have glucoses link up with each other, as we see down below here in this longer molecule. And here is that amino acid. We're going to spend a lot more time with amino acids when we talk about proteins in a little while. But notice they have that alternate pattern going on today in this structure. And we also saw the alternate pattern in the indigestible cellulose. So whether it's that pattern that makes it like that or not, or it's just the chemistry, that is the question. But this does make for a water-resistant covering that has a base of carbohydrate that happens to have amino acids attached on every single one. And what that's going to do is produce certain biological substances, such as the exoskeleton of arthropods. So here you see this insect is molting. What's leaving behind here is its smaller exoskeleton, which is going to be made of chitin. And that, again, is going to be insoluble with water. Water is going to have a very hard time breaking that up. An insect wouldn't survive long if it touched water and instantly dissolved. So it's going to be insoluble in water. And sometimes we take cues from nature, we look at certain materials that nature provides, and what we can do then, figure out the chemistry and then use them to our benefit. So one of the things that we're going to do then is we're going to take the molecular formula and the molecular structure of chitin, use it to our benefit, and in this case here, it happens to be used for a certain type of surgical thread. So a surgical thread that doesn't dissolve in water and holds its place and is biologically sound is going to benefit humankind, and of course closing up wounds is a very important thing. Biomolecule class number 2, we have the lipids. Now, the lipids have a defining characteristic, which is this. They're a diverse group of insoluble molecules. And what that means is they do not readily dissolve in water. So lipids are the only class of large biological molecules that do not form polymers. We mentioned that before, that monomers join other monomers. They form these big, elaborate, long chains of biomolecules. Lipids don't do that. They have a defined set of sizes that we find them in. The unifying feature, we just mentioned it, is that they do not dissolve in water, so that means they have little or no affinity for it. Lipids are hydrophobic. Now, let's break that word down. Hydro, again, we relate that to water. Phobic, we can relate to the word phobia, so they're water-fearing. If you have a phobia, you have a fear of something. Lipids don't like water very much. Why? Well, if you go back to our chemistry of biology talk, they consist mostly of hydrocarbons. And when you have a lot of carbon and you have a lot of hydrogen bonding together, what we form there is very nonpolar covalent bonds that don't get along well with water. So nonpolar covalent is going to be the character of the bonds that you find in the lipid family. Now, there's going to be 5 important lipids that we're going to deal with this year. First is fats, second, phospholipids. Third will be steroids, fourth, oils, and number 5 is going to be waxes. All of these are biological components of living things. So we'll take a look at every one of these types of lipids in the next few slides. First on the list was fats. Fats are constructed from 2 smaller molecules. Let's look at those 2. The first one is going to be called glycerol. And as you can read, a glycerol is a 3-carbon alcohol that has a hydroxyl group attached to each carbon. So what we're looking at here is this red area here, this base where you see these three arms coming from, that is going to be your glycerol base. If we want to get a little more precise on what that is, if we take a look at this molecule here, we have 1, 2, 3 carbons. We do have an alcohol when glycerol is alone that's going to sit off this right side here. But this glycerol is not alone. As you can see, there's 3 arms coming from it. And what those arms are what's called fatty acids. Fatty acids consist of carboxyl group attached to a long carbon skeleton. So if you look at these up-down arrangements here of these atoms, those are carbons, and all these little white guys sitting off here on top and on the bottom, those are all hydrogens. So again, chemistry has that shorthand of drawing molecules. And sitting on the left edge here, we have the carboxyl group. You met the carboxyl group when we were talking about the functional groups of biological molecules. And they typically will have a carbon atom right here, double bonded to an oxygen, single bonded to another oxygen, and when alone, there'll be a hydrogen on the end of it as well. So our question becomes, how do we build a fat? Well, in this case, we see our glycerol molecule here. It's up in the left-hand corner. We have this beige box around it, and we said it had some hydroxyls attached to it. And as you can see here, what's happening is a fatty acid is now coming into the picture, and it's headed towards the glycerol molecule. They're going to bump into each other, and what we'll see is the hydroxyl group from the fatty acid will bond with the hydrogen from this hydroxyl group, and again, water comes out. We've seen that before in the carbohydrate groups when they link up. So with the loss of a water molecule, let's recall what that's called again, and we said it was called a dehydration synthesis reaction. So that would be the first step in building a fat. Now, if you look down here, not only do we see the name of the connection, which is ester linkage, but we also see that it's not just one fatty acid that is attached. We have 2 more there as well. And they went through the same mechanisms to attach the fatty acid to the glycerol molecule. So we have glycerol attached to 1, 2, 3 fatty acids, and that's what we call a fat, or more specifically, a triglyceride molecule. Now, fatty acids can be unique as well. They can vary in length, and that length is going to be based on how many carbon atoms you find in the long chain that comprises it. Also, we're going to see the possibility of what's called a double bond in chemistry exist within these fatty acid chains. Now, the first type of fatty acid chain, the first class of them, is going to be called a saturated fatty acid. And what that means is that every carbon that you find in the fatty acid chain is loaded up with as many things as possible. Now, we had said that carbon has the possibility of bonding to 4 things. And all we have to do to check to see if this thing is saturated is see that each carbon in here is bonded to 4 things. 1, 2, 3, 4. 1, 2, 3, 4, and we see that the whole way down until the end of the molecule. The molecule is absolutely loaded up with hydrogens, and there's no room for anything else to be attached to it. Now, the other type of fatty acid is called an unsaturated fatty acid. And again, to check, all you'd have to do is take a look at the chain and see is every single carbon bonded to 4 different things. And the answer is no. Right here, you see that these 2 carbons are bonded to 1, 2, 3 things and 1, 2, 3 things. And that usually signifies the presence of a double bond. And you can clearly see the double bond right here. That's the other way of looking, just spotting the double bond. So make no mistake there. It's very easy to spot one of these unsaturated fatty acids. But it's not necessarily just one that you're always going to find. You may find more. And that would make a particular type of fatty acid a polyunsaturated fatty acids if you could find more than one double bond within the chain. When you have those saturated fatty acid chains and there's no double bonds present, what we have is a saturated fat at that point. And most saturated fats are going to be solid at room temperature. Another thing about those is that they usually are going to come from animals. So if we take a look at the chemistry of this molecule, and we'll just highlight one of these fatty acid tails right here, notice that they've gone even more short-handed with the way they drew this out. Because this diagram is so small, they didn't bother putting in all the carbons or all the hydrogens. They just put this zigzag line, which signifies a carbon chain. Now, if we take a look at it, you can see there's no double bonds anywhere in here. The shape remains the same along the way. So that means that this is going to be representative of an animal fat, which is going to be saturated. Now, there's also fats that as a whole are considered unsaturated fat, and those tend to be more liquid at room temperature. And those also are going to derive their origins more from plants than from animals. However, we have the exception of fish. Now, if we take a look at the chemistry of this molecule down below, you'll see that in that third fatty acid chain position, you have a different shape. You don't have that perfect zigzag up and down type of structure there, but you have this right in the center. And you see that you have a line going across the top and there's one underneath it. That signifies a double bond, which tells you that one of these fatty acid chains is unsaturated. And how is that going to affect this particular fat? Well, if you look at the space filling model right here, notice that it is now bent. So now it has two straight chains sticking off the side, and one of the legs is bent. And that little leg being bent is going to be enough to resist tight packing of this fat, which we see in, for example, this butter or an animal fat in meat. And instead, it's going to go more of the liquid route because of this little bend right here. Once you have a few billion molecules next to each other, they're going to space a little further apart. The attractive forces between them won't be as strong as if you could fit them in a nice, neat little package. And for that reason, they're actually going to be liquids. What can be surprising to some is actually the function of fat, and it winds up being energy storage. Fat actually has over twice the amount of energy potential as a carbohydrate or a protein. So animals that have abundant fat stores will actually have will have more long-term energy than one that has a lower body fat percentage. A certain type of cell will carry this fat, and that's called an adipose cell, and we'll deal more with that when we get into tissues later this semester. But fats, as a whole, are going to cushion vital organs, so we don't want them ramming up against each other with nothing in between them because they can be damaged, and they could also insulate the body. So if you have animals that live in warm-blooded animals that live in cold climates, what you're going to see is they're going to have a higher percent of fat storage capability than one that live in the tropics. Next up, phospholipids. And as you can see here, it has a slightly different shape than we saw with the triglyceride or the 3 fatty acid fat from the slide previous. Now, in a phospholipid, instead of 3 fatty acid tails, there's going to be 2. So if you look at the top, not only do we have glycerol present, but we also have a phosphate group and we have a choline sitting on top. So here is the chemistry, the structural formula of all the atoms that make up what we call the head of the phospholipid. Now, the fatty acid tails, we can see that they're found below, and those fatty acid tails are very much like what we saw with the fats, and the fact that they're long carbon chains. You can see them here, and you can see some of them have that double bond we were talking about, meaning that that particular fatty acid chain is unsaturated. Now, a very shorthand way to draw it when your diagram is full of phospholipids is like this. They just draw a head, and they draw a couple of little bent legs coming off of it. And that's kind of symbolism telling me that the diagram I'm looking at is trying to represent the phospholipid without going this far in detail. Here we have a diagram of phospholipids, hundreds, even thousands of phospholipids, and we see how they self-assemble themselves in a watery environment. Notice that you have water on both the outside and the inside of the two sides of this particular structure. This structure is going to be called a lipid bilayer. And what I want you to notice is on the inside, all those hydrophobic hydrocarbon legs are pointed towards the middle. Even on the other side, they're kind of upside down. They're going towards the middle as well. And the reason is they're made of hydrocarbons. And we said hydrocarbons hate water. They're hydrophobic. They fear it. So what they can do, though, is they can mingle with each other without any problem. So they form this waterproof barrier in the center where the hydrophilic or water-loving heads sit on the outside and they come in contact with the water. That is how lipid bilayers self-assemble themselves. And this is going to be the characteristic structure of all cell membranes. So on the outside of cell membranes of all living organisms, we're going to find this structure. Also on the interior of cells within the cytoplasm, we have these things called organelles, many of them made up of membranes as well, and they're going to have the exact same phospholipid structure for their lipid bilayers. Our next lipid type is going to be the steroids. They're characterized by a carbon skeleton that has 4 fused rings. So those fused rings are right here, 1, 2, 3, and 4. Now, what differs on these steroids is going to be what we call the R group, which is going to be this group of atoms up there. With a different R group present, that means you have a different steroid. Now, the most famous steroid that we study in biology is going to be cholesterol. Cholesterol is absolutely necessary because you're going to find it in every one of those membranes we were just talking about. However, too much cholesterol can be bad. Why? Because it can build up on the interior of vessel walls, and if it breaks out and plugs up that vessel, it can lead to things like myocardial infarction, which is a heart attack, or possibly a stroke if it happens in the head as well. So another 2 steroids that are connected to cholesterol are our male and female hormones. So do we know what those hormones are? First of all, think of a male hormone. Can you think of any? Well, testosterone is going to be the preeminent male hormone, whereas estrogen is going to be a female hormone, and they're going to be both based off of this cholesterol molecule. We also have waxes. Waxes is going to be when you have a fatty acid that's connected to an alcohol, and both of these tend to be fairly long-chained. What they're going to do for an organism biologically is give it a protective covering by way of a secretion that will resist water and degradation. Now, as you can see here, if we take a look at the chemistry of it very simply, we have a fatty acid connected to a long-chain alcohol. Well, what might that look like? Well, here's a long-chain fatty acid, here's its carboxyl end, and here's our long-chain alcohol with its hydroxyl end, and it meets kind of in the center. That could be the basis for a wax. Now, what do waxes do besides resist water and degradation? Well, they also provide some protection as well. Let's take earwax for an example. If we look at it scientifically, we're going to call it ceramin, and it has the protective function of being bactericidal, fungicidal, and possibly even antiviral. So where we have holes in our body, we have to have mechanisms that will stop any invaders that want to do us harm by getting in those holes. In our ears, for example, we have wax there to protect our auditory sense, keep it safe, and keep those harmful things out. I'm going to make this a stopping point of this screencast for biomolecules. So far we covered basic mechanisms of biomolecules, how they're built, how they're broken down. We then went into the family of carbohydrates, and then we went into the next group of biomolecules called the lipids. In part 2, we'll be taking a look at proteins, and then we'll take a look at nucleic acids, and we'll do a final summary.